Now let me take you into TCA cycle. Two case stems that I have given here related to TCA cycle. There are a couple of TCA cycle enzymes, high yield enzymes. We will go over what is the yield point for each one of them. So the first one here, a 2-year-old boy is being examined in the emergency department following a period of vomiting and a seizure. Physical examination finds that infant has significant hypotonia, drooping eyelids, limited eye mobility. Molecular studies carried out on muscle biopsy cells find that the coenzyme binding site of one of the subunit of succinate dehydrogenase has sustained deletion. See, I did not mark anything so far except succinate dehydrogenase. So what I do now in my mind, I am focused on succinate dehydrogenase, nothing else. I forgot all other enzymes right now. I am only with succinate dehydrogenase. Okay, so this enzyme has sustained a deletion. That means there is a deletion of gene for this enzyme. That's it. That means it is not available, right? Given these results, which of the following is most likely no longer capable of participating in the function of the enzyme? So indirectly what this question is asking, what is the function of succinate dehydrogenase? If you ask me, Dr. Mungli, can you make a single line question on this? That's my question. What is the function of succinate dehydrogenase? That's it. That is what they're asking. What is the function of succinate dehydrogenase that you have studied so far? Anywhere you came across this name, succinate dehydrogenase? Complex 2. Remember complex 2? There are 3 names for complex 2. Succinate dehydrogenase, which is in TCA cycle, and the other is ETF, electron transferring flavoprotein, and the third is glycerol phosphate dehydrogenase. So this question is related to complex 2 function of succinate dehydrogenase, which is also part of TCA cycle. Okay, that's how you need to think on that. Next one. A scientist has developed a drug that when added to eukaryotic cells lead to elevated lactate levels. See, there is a lactic acidosis here. Analysis of mitochondrial content also demonstrated elevated alpha ketoglutarate levels. Alpha ketoglutarate is also elevated. This drug may be interfering with the reaction that requires which one of the following vitamins? What do you think? What you are looking at, like if you see 2 things elevated here, there are elevation of lactate, elevation of alpha ketoglutarate. What you are thinking? Which enzyme? What is the enzyme? What is the general enzyme that is kind of maybe problematic here? Complex enzymes. Remember those 3 complex enzymes? If there is a mutation in E3 gene subunit, then you will have elevation of lactate, elevation of alpha ketoglutarate, elevation of branched chain alpha keto acids. So this question is related to those 3 complex enzymes because they have mentioned only 2 elevations, but you will figure it out. It's kind of okay given information to take you that side. So it's about complex enzymes. And what are the coenzyme requirements for those complex enzymes? How many coenzymes? 5 coenzymes. And you should know what all those 5 coenzymes. Vitamin B1, B2, B3, B5, lipoic acid. Any one of these vitamins deficiency can be the answer here. Is it not? That's how you will figure out the correct choice. So now let's look into TCA cycle. This is just an overview of TCA cycle here. Focus on the products that is written down there. Look at the products. Each cycle, it is written for each cycle. 1 acetyl-CoA, as you see, acetyl-CoA, your acetyl-CoA from the previous reaction, it is now getting into TCA cycle. And how many carbons in acetyl-CoA? 2 carbons. And look at the products. How many carbon dioxide released? 2 carbon dioxide released. So the 2 carbons in acetyl-CoA are entirely removed now. So it is a complete oxidation of acetyl-CoA. TCA cycle completely oxidized acetyl-CoA. That's the first thing you got to know. Then what is the outcome of this? What is the benefit for the cell? If you are oxidizing acetyl-CoA completely, cell has to get some benefit. You are here in the class. What is the benefit? In the class, you came to class today. What is the benefit? You'll get some marks in the exam. You study, you learn, and you take it, right? And that's what I should try to do. I should, justify your time. You are spending 2 hours of your lifetime with me today. Is it not? 2 hours, it is your lifetime. You are spending that with me, and I am spending my 2 hours of lifetime with you. We don't actually think like that. Can we take this 2 hours back? Impossible. No one can give you back except him. Right? See, it is such a thing actually. If you seriously look at it, every hour that you get, that's your lifetime. So you got to make use of it. So whenever I come to class, I always think that way, that 2 hours that you are spending with me, I should make it worthwhile to my maximum, ability, skills, capacity, whatever it is. So that when you go out of this class, you felt I learned something. Is it not? Otherwise, it's waste. If you would have studied on your own, right? Why to come here? Anyway, so that is my point. That's how I look at it. So acetyl-CoA, 2 carbons, entirely breaks into carbon dioxide. That is completely oxidized. And this carbon dioxide, we throw it out, exhale it out. Okay. And then what is the benefit for the cell? The cell will get these 3 things. It's going to get 1 ATP in the form of GTP. In fact, what it gets is GTP, not ATP. But we measure it in the form of ATP in terms of energy. Otherwise, the product is GTP. I'm going to come to that point later. What is the use of GTP? And then you're going to get NADH. How many NADH? You get 3 NADH. And each NADH is 2.5. 3 NADH is how much? 2.5, 2.5, 7.5. Right? And then you get 1 FADH₂, that is 1.5. So if you count them together, how many ATP equivalent? So you're going to get 10 ATPs, is it not? 10 ATPs, 3 NADH, 1 FADH and 1 ATP, it is equivalent to 10 ATPs. That is why when acetyl-CoA is oxidized in a cell, the cell will get 10 ATP. That is the benefit for the cell. Okay. So of course, NADH has to go into electron transport chain, FADH also has to go into complex 2 in the electron transport chain, and then it will get ATPs, which we have seen already. Okay, so with that understanding, let's look at these names here. How acetyl-CoA is undergoing oxidation into carbon dioxide. Let me now focus on that. So before I focus on high yield enzymes, let me just explain you overall, and then we will go into individual enzymes. So we have citrate synthase, which will combine acetyl-CoA with oxaloacetate to make citrate. And then citrate undergoes further oxidation process to become isocitrate done by aconitase enzyme. And then isocitrate dehydrogenase converts that into alpha ketoglutarate. And then alpha ketoglutarate dehydrogenase complex is missing here. So converts alpha ketoglutarate into succinyl-CoA. And then succinyl-CoA to succinate done by succinyl-CoA synthetase. And then succinate to fumarate, succinate dehydrogenase, fumarate to malate, fumarase enzyme, malate to oxaloacetate, malate dehydrogenase. So there are 8 enzymes in a TCA cycle participating in breakdown of acetyl-CoA into carbon dioxide. Out of these 8 enzymes, okay, before that, look at oxaloacetate. Oxaloacetate here, which is a 4-carbon molecule. Acetyl-CoA is a 2-carbon, and these are the 2 carbons which we are releasing. So in the very first reaction, you are combining acetyl-CoA with oxaloacetate to make citrate. And citrate is, that means this citrate is 6-carbon molecule. Is it not? Like 2 carbon plus 4, you got 6-carbon citrate. And this citrate, the 6-carbon citrate, just to write, you don't write down this, whatever I'm writing here. I just want to explain you something. So 1, 2, 3, 4, 5, and 6 carbons. In that, citrate will have carboxyl group like this. I'm just focusing on carboxyl group. Just to tell you something. There will be 3 carboxyl group in the citrate. 3 carboxyl groups will be there in citrate. And that's why it is called TCA cycle. What is TCA? Tricarboxylic acid cycle. Tricarboxylic acid is nothing but citrate or citric acid. Citrate is a tricarboxylic acid. It has 3 carboxyl group like this. That is why the name. Just in case, if you are wondering what is TCA, why it is called TCA, this is why it is called TCA. So TCA cycle, and in the middle, see what they have written for TCA cycle within the figure. It is written Krebs cycle. See, it is written Krebs cycle, which is TCA cycle, and also it can be written as citric acid cycle. Citric acid cycle. Citric acid cycle, because citrate is a deprotonated form of citric acid. So citric acid cycle. These are the just names which are used interchangeably. So whenever you come across these names, just know that it is all same. Okay. Anyway, with that, see what happened with oxaloacetate. Oxaloacetate, after combining with acetyl-CoA to make citrate, and when all this oxidation going on, right, there will be release of carbon dioxide, 2 carbon dioxide released. And the rest of the carbons that are present in this succinyl-CoA, as you see here, succinyl-CoA has got 4 carbons remaining, because 2 carbons you have released as carbon dioxide by this time. And those 4 carbons, eventually, they will go and reform oxaloacetate again. My point here is, in TCA cycle, oxaloacetate is not consumed. Is it not? It is remade every time. Every time it is regenerated. What is being oxidized is acetyl-CoA only. That is the point. General point. Not for exam. Just for your understanding. Only acetyl-CoA is oxidized in the TCA cycle. Nothing else. Okay? That is the point. So with that understanding, let's see the high yield enzymes. That is what will be asked in your exam. High yield enzymes. You want to break before we get in this? Let's take a break, and I'll start at maybe 9-ish, 9:09 to 9:05. By that time, you please come back. Okay. All right. On the side that I have I have written oxidation of glucose, as you can see. Glucose, which is a 6-carbon molecule, I'm going to write that here, 6-carbon, undergoing glycolysis. So this is in the cytoplasm. This is a cytoplasmic thing, cytoplasm, glycolysis. And you get 2 pyruvates. And the net number of ATPs and NADH that you are getting here, net ATP is 2 from glucose to 2 pyruvate, and you get 2 NADH. And each NADH is 2.5 plus 2.5. So that is 5 ATP, right? From 2 NADH, you get 5 ATP, right? So 2 ATP plus these 5 ATP. So let's keep those numbers just to see how many ATPs we get. So, and then these 2 pyruvate has to go into mitochondrial matrix. This is the matrix here, matrix of mitochondria. Matrix of mitochondria. Rest all happens in the matrix of mitochondria. 2 pyruvate converted into 2 acetyl-CoA. So you're going to release 2 carbon dioxide and again make 2 NADH plus, which is worth 5 ATP. See, from 1 glucose, by the time you make 2 acetyl-CoAs, already you have released 2 carbon dioxide and you have made 2 plus 5 plus 5. So 12 ATPs you have made so far. Is it not? Right? 12 ATPs we got. In the glycolysis, you got 7 ATPs and 2 pyruvate to 2 acetyl-CoAs, you got 5 ATPs there. So total we got 12 ATPs so far. And then these 2 acetyl-CoAs enters into TCA cycle. Here is your TCA cycle. TCA cycle. Each acetyl-CoA is oxidized into 2 carbon dioxide. So 2 acetyl-CoAs means 4 carbon dioxide. And each acetyl-CoA gives you 10 ATPs. I have shown you like it is there on the side here. And 2 acetyl-CoA means 10 plus 10, 20 ATPs. So if you count those 10 plus 10 here, so and count all of them, you're going to get 32 ATPs. 32 ATP. That is the complete oxidation of glucose to give 32 net ATPs. Okay, this is how cell will make ATPs by oxidizing glucose all the way into carbon dioxide. How many carbon dioxide? 6 carbon dioxide. 2 in the pyruvate dehydrogenase complex, 4 in the TCA cycle. So if you carefully look at it, the glycolysis in the cytoplasm, TCA cycle in the mitochondrial matrix. Who is going to connect glycolysis and TCA cycle together? Which reaction is a connector reaction between glycolysis and TCA cycle? Pyruvate dehydrogenase complex. That is a connector reaction, connection. The connection between glycolysis and TCA cycle is pyruvate dehydrogenase reaction, which is converting pyruvate into acetyl-CoA. Okay. All right. So just wanted to tell you that. And also one more thing, the reactions when they are happening in the cytoplasm or happening in the mitochondria, all these enzymes except one enzyme, which I'm going to tell you now in a couple of minutes, they are all freely floating enzyme in the cytoplasm. Remember, they are not fixed anywhere. They like cytoplasm is like an ocean. That Caribbean Ocean that you're seeing down there. It's like that. In our cell, cytoplasm is like that. How many fish in that ocean? Yeah, that's the best answer. Lot. You don't know. It can be millions, billions. We don't know how many. So let's consider all those fish as enzymes. Each fish is an enzyme. Let's consider that way. And that fish eat food. That is their substrate. Right? So these enzymes, they are freely floating. They don't know where is their substrate. They got to find those substrate. That substrate has to come to this enzyme, or this enzyme has to go there by chance. And when it goes there, bind to the active site, then it will make a product. And that product, again, it is a freely floating, finding its enzyme, because the product of one enzyme is a substrate for another enzyme. Is it not? Like in the cycle. And that enzyme has to find this product, which is going to be a substrate for it. Like, just imagine. Is it not wonderful how this body functions as a union, one unit, trillions of cells functioning together, and there is so much randomness inside. It's a chaos inside. Imagine like billions or millions of enzymes, substrate, everything is packed in the cytoplasm and reaction has to go on. It's not like what you see as a cycle. You don't see TCA cycle round, round, round in the cytoplasm or matrix of mitochondria. You don't see that. It's all randomness there, chaotic. So order has to be there in that randomness. And that is what is our cell is. Anyway, so the most high yield reaction means let's go into the high yield reaction. You are waiting for that. So let's go to that. I have written all those things down in the notes. If you miss anything, don't worry. It is there in the notes. I hope you have that notes. You see the notes? PDF file? Yeah, she said no. Kitana, which side you are downloading? Is it a PDF file? No notes? Do you see notes in PDF file? Okay. No, it is important. I want you to have this notes. So look at this. These are enzymes with what is the importance of each of these, but I will highlight the important ones. So the important ones are aconitase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase complex, succinyl-CoA synthetase, which is also called as succinate thiokinase. I will tell you later. Succinate dehydrogenase and malate dehydrogenase. These are the important enzymes. So out of 8 enzymes, 6 enzymes are important. Okay, each one of them, they have their own importance. So the first enzyme, aconitase, which is converting citrate to isocitrate, importance is rat poison, fluoroacetate has got a effect on aconitase. It's a non-competitive inhibitor. Non-competitive inhibition on aconitase enzyme. So that's what I written down there. What happens is this fluoroacetate, it will combine with citrate to become fluorocitrate. So I'm going to write down here, fluoroacetate become fluorocitrate. And this fluorocitrate is the one that will have a negative effect on the enzyme. So the non-competitive irreversible inhibition on aconitase is actually done by fluorocitrate, not fluoroacetate. Fluoroacetate is a rat poison. That's the active ingredient in that rat poison. And when that poison gets into the cell, that fluoroacetate becomes fluorocitrate inside. And that fluorocitrate irreversibly go and bind to aconitase enzyme, and then it's going to inhibit the enzyme. And that is how the citric acid cycle or TCA cycle is blocked. That's how rat will die. That is the mechanism of rat poison. How to kill a rat? How to kill a rat? Use rat poison or use that case thing, right? But they will run away. When you open, they will run. How many of you did this? You know that small little case? So, and they are very intelligent rats. They don't get in there. They know there is something wrong with this. They are not going to do it. Even rat poison also, they don't eat because they have strong sense of smell. They know inside there is something. Even if you keep it in the butter, to attract, inside butter you kept rat poison. And they smell that what is inside. Most of them, they don't eat. Those who are unfortunate rats, they will eat and die. Okay. So anyway, why I am saying this to you? Some people, they will consume rat poison to die, suicide. And that's where you come in as an emergency doctor. That is why we need to know this. Okay. So what you do, you wash the stomach lavage, stomach washed, take out what is not absorbed and manage symptomatically, because eventually it will go out of the body in the urine. Hydration urine, put it out. Okay. That is what is the importance of, that's how TCA cycle affects our cells when a person consumes rat poison. Okay. So coming to the second important reaction, isocitrate dehydrogenase. Why this reaction is important? Because isocitrate dehydrogenase is most regulated enzyme in the pathway. Okay, regulatory molecules, it is there in the next slide. Going to see that. And also in this reaction, carbon dioxide is released. One of the carbon of acetyl-CoA released and NADH is produced. NADH is made here. And that's why sometimes if the question is related to which reaction in TCA cycle that makes NADH or which reaction in TCA cycle that makes carbon dioxide, if that is what is the question, directly or indirectly, this is one of the enzyme, isocitrate dehydrogenase. Otherwise, majority of time, the question on this enzyme will be on regulation, because this is the most regulated enzyme in the TCA cycle, just like your PFK1. So, entire TCA cycle, if you control this enzyme, you control TCA cycle, the phase, the fastness of the TCA cycle. Okay, coming to the third one. Third important enzyme is alpha ketoglutarate dehydrogenase complex, which converts alpha ketoglutarate into succinyl-CoA. So why this reaction is important? It is because this reaction is inhibited by arsenate. Arsenate inhibits this reaction. Even PDC also inhibited by arsenate. We are going to see that later. Arsenate inhibit this reaction. It needs 5 coenzymes, just like any other complex enzyme, 5 coenzymes. Remember those 5 coenzymes, TPP, B1, B2, B3, B5, and lipoic acid, all that. So, carbon dioxide produced, carbon dioxide released here. That's the second carbon of your acetyl-CoA, and NADH made. Okay, so this makes it as one of the important reaction in TCA cycle for exam. So, alpha ketoglutarate dehydrogenase complex converts alpha ketoglutarate to succinyl-CoA. The importance of this enzyme is arsenate inhibit this enzyme. 5 coenzymes are necessary. Carbon dioxide released, NADH made. Okay, coming to the succinyl-CoA synthetase. Succinyl-CoA synthetase is also called as succinate thiokinase. Succinate thiokinase. Thiokinase. What is the importance of this reaction is it is GTP is made here. GTP is made and example for substrate level phosphorylation. This is an example for substrate level phosphorylation. Because you are making GTP, which is like ATP, energy rich molecule, without going into electron transport chain. So this is an example for substrate level phosphorylation in TCA cycle. These 2 points make this reaction important for exam. Succinyl-CoA to succinate is the reaction. Enzyme name is succinyl-CoA synthetase. Okay, and other name for that is succinate thiokinase. And where GDP enter into reaction and GTP is made. So GTP is made here. And substrate level phosphorylation. This is an example for substrate level phosphorylation. So the thing is, wherever GTP is necessary in our cells, because GTP is made here, wherever GTP is used, this can be one of the source. That means you need to know where GTPs are used. Any examples where that you have studied, you know, where GTPs are used. GTP is being used. Whenever you come across GTP, anywhere so far, starting from first semester. Second messenger, remember adenylate cyclase to CKMP, all that alpha subunit, G protein coupled receptor, alpha subunit where GDP is replaced by GTP, alpha subunit is active. From where you get that GTP, this reaction. So G protein coupled reaction, GTP can come from this reaction also. that is succinate thiokinase reaction or succinyl-CoA synthetase. So it can be connected that way. So I will write in simple words, G protein coupled, I'll just write it as G protein. So you understand what I meant, okay? G protein. So G protein and alpha subunit GTP, or you can write it that way, alpha GTP, alpha subunit GTP, G protein. That's where you need GTP. And then another reaction which we are going to study is gluconeogenesis. Gluconeogenesis. In gluconeogenesis also you need GTP. This can be one of the source of GTP. So, and later wherever you come across GTP, you can, you just know that GTP can come from this reaction. That's how you can connect. Okay. So that is the importance of this reaction here. It can be connected that way. It can be connected to G protein coupled reactions, can be connected to gluconeogenesis in an integrated question. TCA cycle with gluconeogenesis. Okay. All right. Coming to our next high yield reaction is conversion of succinate into fumarate by succinate dehydrogenase. And why this enzyme is important? Because this is complex 2. This enzyme is complex 2 in ATC. This is complex 2 in ATC. That's why this is an important enzyme. Anything wrong with this enzyme, complex 2 is affected. That means your ATC is affected. Okay, that's how you can connect. So TCA cycle connection with ATC through succinate dehydrogenase. And as you see, there is a production of FADH₂ here and one FADH₂. So FADH₂ is made. FADH₂ made, and which is worth 1.5 ATP. All right. Next reaction that is important is conversion of malate into oxaloacetate done by malate dehydrogenase. And the importance of this enzyme is just that NADH is made here. That's it. NADH made. That's it. These are the importance of each one of these high yield reactions. So there are 6 high yield reactions. Each one of them have their own points for you to remember. So what did I tell you? Each high yield enzyme, you need to know the substrate and the product. Remember that. Because sometimes in the exam, like especially in the choices, they will write substrate and product. Instead of writing enzyme, they write substrate and product, substrate product. Let me give you an example. I meant to ask, I want to connect TCA cycle with gluconeogenesis. That's in my mind. While making question, I want to make an integrated question. I want to connect TCA cycle with gluconeogenesis. And I made a stem, so and so, whatever. I made it. And then I'm asking, instead of asking which enzyme is providing the GTP required, means energy required for gluconeogenesis process, the specific reaction I'll mention is coming from which one of the following reaction. Instead of writing which one of the following enzyme, I wrote which one of the following reaction. So then I'll start writing citrate to isocitrate, isocitrate to alpha ketoglutarate, alpha ketoglutarate to succinyl-CoA, succinyl-CoA to succinate, succinate to fumarate. I'll just go on writing that A, B, C, D, E. So your answer is succinyl-CoA to succinate. So that's how sometimes you may see that. I don't see repeatedly that kind of where substrate and product is mentioned, but it doesn't mean that they don't ask. They means me and somebody else also. Okay. So mostly we ask enzymes, mostly, but that is also possible. Sometimes we do that. Okay. So that is why it is better to know substrate and product for each high yield enzymes. So that's about high yield enzymes. Any question here with any of these enzymes? Also go through the notes, see if there is anything missing. Most of it I have written. Okay, and also highlight those 6 enzymes because I wrote all 8 of them. Just highlight 6 of them. That's it. Okay, so now let me take you into regulation. Regulation is Simple. You need to know one enzyme here. Isocitrate dehydrogenase, master controller of TCA cycle, just like your PFK1. You control that enzyme, you control the pace of TCA cycle. That's it. So look at the positive modulators. Already I explained the role of calcium and ADP with PDC. You know, pyruvate dehydrogenase complex, whenever there is a lack of energy, whenever you are exercising, you know, ADP and calcium, what they do? They keep your PDC high and convert pyruvate to acetyl-CoA, and that acetyl-CoA has to go into TCA cycle in a faster way. And that is why same calcium and ADP, which were positive modulators on PDC, same molecules will have a positive effect on isocitrate dehydrogenase. That's it. You don't need to worry about anything else. There is no extra information here. It is just another enzyme. Whatever the explanation that is there for PDC, for calcium and ADP as a positive modulator, it is same for TCA also. Only thing is enzyme is different. That is isocitrate dehydrogenase. Okay. So that is the point. Coming to NADH. What is the role of NADH on TCA cycle? Negative. Because NADH is the product of TCA cycle. Is it not? NADH, like see, products it is written. NADH is also written here. NADH, whenever there is a lot of NADH, when NADH is not being oxidized. Example, your electron transport chain is down. So NADH is not being oxidized. So they will build up. When there is a buildup of NADH, for whatever the reason, they will have a negative effect on isocitrate dehydrogenase. That is the key enzyme. NADH will have a negative effect on isocitrate dehydrogenase, thereby slows the TCA cycle. Because when you have plenty of NADH, why to run TCA cycle so fast? You slow it down. So TCA cycle slows down. Because you know NADH is made in TCA cycle by 3 enzymes. One is isocitrate dehydrogenase. Other is, see, look at this, alpha ketoglutarate dehydrogenase complex also makes NADH. Malate dehydrogenase also makes NADH. So these 2 enzymes also will be negatively modulated by NADH. Feedback negative effect on their own enzymes. So NADH, not only it has got a negative effect on isocitrate dehydrogenase, it also has a negative effect on other 2 enzymes which are making it. That is alpha ketoglutarate dehydrogenase complex and malate dehydrogenase. Okay. So that's how TCA cycle is regulated. ADP and calcium positive on isocitrate dehydrogenase. NADH is negative on isocitrate dehydrogenase, of course. Along with that, NADH has negative effect on other 2 enzymes as well. Okay. So, but most importantly, it is isocitrate dehydrogenase because that's the key enzyme. All right. So that's about regulation of TCA cycle. Coming to arsenate toxicity. Arsenate, it inhibits both PDC and TCA cycle. Pyruvate dehydrogenase complex, it is a complex enzyme. It has E1, E2, E3 subunits and E2 subunit. I told you it needs lipoic acid and pantothenate. And the lipoic acid, I told you it has SH group, thiol group. And this arsenate will go and bind to the thiol group of lipoic acid in E2 subunit. Okay, and that's how it's going to inhibit the complex enzymes. There are 3 complex enzymes. Arsenate will inhibit all 3 of them. Here I have written 2, PDC and TCA cycle. In TCA cycle, what is your enzyme which is inhibited by arsenate, which you have written down in the notes? Alpha ketoglutarate dehydrogenase complex. So that is the enzyme in TCA cycle which is inhibited by arsenate. So I'll write it in the bracket. In TCA cycle, it is alpha ketoglutarate dehydrogenase complex That is the enzyme which is inhibited by arsenate. So, and where exactly arsenate binds to this enzyme? E2 subunit. Remember that. That can be your choice. E2 subunit. Which part of E2 subunit can also be your choice? What is that? Lipoic acid. Lipoic acid. Why? Because it has SH group, thiol group. Arsenate will go and bind to thiol group. Okay. The first statement. Blue color. Look at that. Important. And then, because it is inhibiting pyruvate dehydrogenase complex, so pyruvate is not oxidized to acetyl-CoA. That means pyruvate goes into lactate. So there is a lactic acidosis. Lactic acidemia means lactic acidosis. Okay, so accumulation of alpha keto acids, other two alpha keto acids also you can see, because those enzymes are also inhibited, because they also have E2 subunit, lipoic acid. So that means patient will show elevation of alpha ketoglutarate, branched chain alpha keto acids, overall giving rise to metabolic acidosis. It is just like, you block E3 gene and then all these things are elevated, something like that. Here also, you are blocking all the three enzyme activity by attaching arsenate to E2 subunit. All the three enzymes are down, leading to metabolic acidosis. How do you recognize arsenate poisoning or arsenate toxicity? What are the physical signs and also other signs? So look at the signs and symptoms here. Very characteristics and interesting and easy to remember. Look at the figures. See the nails, ladies. Look at the nails. Maybe you think about getting them next time when you go for a semester break. Look at them. Is it not interesting? Bands. Okay, that's how there will be coloration on the nails of arsenate toxicity on a person or a patient. So abdominal pain, raindrop pigmentation, I'll explain that, hyperkeratosis on the palms and soles, and white lines on the nails that are called as Mies lines. Then whatever you are seeing on the nails, it is called as Mies lines. Alternate pattern of, white lines, white and pink, white, pink, like that. And that is a characteristic of arsenate toxicity. Okay, so Mies lines. And then raindrop pigmentation. Look at this figure here. There is a hypopigmented area and surrounded by hyperpigmented area. You see here. Hypo and hyper. Hypopigmented area and surrounded by hyperpigmented area. That is called raindrop pigmentation. Like when, when rain drops onto the ground, freshly made ground or something with a lot of dust, and raindrops falls, and there is drop here and there, and that's how it looks like. Okay, raindrop pigmentation, very characteristic. Hyperkeratosis on the palms and soles, and that is in the second figure, this figure. If you feel their hand, it is rough hyperkeratosis. So hyperkeratosis on palms and soles, raindrop pigmentation, Mies lines, and garlic odor to breath. All of them will give you a clue to arsenate toxicity. Very interesting. Even if there is one sign, you should be able to diagnose, is it not? Raindrop pigmentation, it is arsenate. Garlic odor, it is arsenate. Mies line, arsenate. Hyperkeratosis of palms and soles, arsenate. There are very few conditions where you see hyperkeratosis of palms and soles. Okay, so that means what? Should not miss these questions in the exam. Very easy. Once you know it is arsenate, you need to remember enzymes. What is the enzyme? That is also easy. Complex. That's it. Any enzyme that ends as complex is your enzyme. That's it. Even if you don't know what is the thing before that, but complex is there. That is your answer. Can be pyruvate dehydrogenase complex, alpha ketoglutarate dehydrogenase complex, branched chain alpha keto acid dehydrogenase complex. And where exactly arsenate binds? E2, lipoic acid. These are the points. All right. Let's do question. All of you, take out your mobiles. Okay. So what is the toxin here? What is the poison? Arsenate. So because there is a garlic odor, arsenate. That's why I just shaded arsenate, which was written in the stem. Without that also, now you know it is arsenate. Okay. So arsenate. So arsenate inhibits pyruvate dehydrogenase complex. In this place, in option D, it could have been alpha ketoglutarate dehydrogenase complex. It could have been branched chain alpha keto acid dehydrogenase complex. Any of those three can be answered here. And also furthermore, if I don't want to write it this way, I'll write E2 subunit of pyruvate dehydrogenase or E2 subunit of complex enzymes or lipoic acid of E2 subunit. That can also be answered. Okay. So option D is correct. Coming to the next one. So what is your answer? Succinate dehydrogenase connected with riboflavin, FADH₂, riboflavin. So answer is riboflavin. Succinate dehydrogenase is right. Why? Ascorbate, biotin, lipoic acid. Nice. Look at that. Each one of you went for that. One of you went for each one of those choices. Other than C. Succinate, whenever you come across this enzyme, succinate dehydrogenase, remember two things. One is complex 2. Other is riboflavin, FADH₂, FADH₂, riboflavin. These are the two things. Okay. Riboflavin as a vitamin, it is needed wherever you come across FMN and FADs. Flavin mononucleotide, flavin adenine, wherever you come across those. Wherever you come across the term flavin, that's riboflavin. Like electron transferring flavoprotein, remember that beta oxidation ETF complex 2? That's riboflavin. Look for flavin, riboflavin. Okay. So succinate dehydrogenase. So what if this question was modified? I'm going to modify that question now. Okay, I'm going to change this. Which of the following is most likely no longer capable of participating in energy generation? I'll just make it like this. In the energy generation. And then here I will just change the choice. Complex 1, complex 2, like that. I'll just go on writing 3, 4, 5. That's it. So answer is complex 2. Succinate dehydrogenase is complex 2. Like that. Then, okay, next question. So if you see elevation of lactate, elevation of alpha ketoglutarate. So what you are thinking about? Complex enzyme, right? And complex enzyme needs how many coenzymes? 5 coenzymes. Because when you talk about vitamins, it's a coenzyme function. So The question is, this drug may be interfering with the reaction that requires which one of the following vitamins? You just need to find out, is there any one of those 5 vitamins in the list? That's it, right? So you don't find biotin in complex, you don't find vitamin K, you don't find ascorbate, you don't find pyridoxine, but what you find is pantothenate, coenzyme A. Okay, that's why answer is C. All right, coming to TCA cycle as a open cycle. This slide is, it looks very simple, but there can be, integrated questions from this slide later on. Make sure you know this slide, star mark this slide. And whatever I'm going to write, you write it down. Later, this slide will be helpful to you, especially in block 2 and block 3, when we do integrated questions. Because this cycle integrates other metabolic pathways, other things that are going on in the cell. Because TCA cycle is referred as an open cycle in the sense that whatever the intermediate is there in the TCA cycle can be removed from the TCA cycle and directed to some other metabolic pathway. Like example, citrate. Citrate is a part of TCA cycle, acetyl-CoA, oxaloacetate, citrate. Citrate can be pulled out of mitochondria into the cytoplasm. And in the cytoplasm, citrate is the one that goes to fatty acid synthesis. Like whenever you eat excess carbohydrate, I told you, in no time it becomes fatty acid. Because carbohydrate glucose converted to pyruvate, pyruvate to acetyl-CoA, acetyl-CoA to citrate. And when the TCA cycle is saturated, when the person is, well fed and resting, that means why do you run TCA cycle if you are resting? If you are eating food, watching TV, why do you need energy? Why do you need energy? You have a lot of food and eating TV. No, sorry, a lot of food and watching TV. Why do you need energy? What happens to the TCA cycle? TCA cycle slows down because your respiratory rate also slows down. Why? You don't need energy. So NADH builds up. And that NADH will have a negative effect on 3 enzymes, remember. So that means eventually citrate builds up. And this citrate, it is pulled out of mitochondria into cytoplasm and go into fatty acid synthesis, triglyceride synthesis, and eventually it will go to adipose tissue. And that is how citrate is connected to fatty acid synthesis and triglycerides, triacylglycerol, and thereby going into adipose tissue. And that's how it contributes to adipose tissue mass. Okay, that is how citrate is being pulled out of TCA cycle. So mark that citrate, fatty acid, triglycerides, adipose tissue. Now, alpha ketoglutarate, our next molecule, alpha ketoglutarate. Alpha ketoglutarate is converted to glutamate. Amino acid glutamate is coming from alpha ketoglutarate. That's why it is a non-essential amino acid. You don't see that in your list of essential amino acids. You know the mnemonic for essential amino acids? What is the mnemonic for essential amino acid? TV till PM, right? You don't see this glutamate in TV till PM because it is non-essential. We can make it. How? Like this. Alpha ketoglutarate, TCA cycle intermediate, can be converted to glutamate. And once glutamate is made, this glutamate can be converted to GABA. What is GABA? Gamma amino butyric acid, which is a inhibitory neurotransmitter. So this is a neurotransmitter, inhibitory neurotransmitter. So that's how TCA cycle is connected with neurotransmitter synthesis through glutamate. And glutamate comes from alpha ketoglutarate. So that's why it can be connected later in amino acid metabolism. And then succinyl-CoA. Succinyl-CoA, it will combine with amino acid glycine. Succinyl-CoA combines with glycine through an enzyme called ala-synthase. And make heme. And heme go into hemoglobin or heme proteins. Wherever heme is required, this is how heme is synthesized. succinyl-CoA, which is a TCA cycle intermediate. It will be pulled out of TCA cycle, combining with glycine to make ALA initially and eventually goes to heme. So there is a pathway for that, and the very first enzyme in that heme synthesis pathway is ALA synthase. And what is the vitamin that is required for ALA synthase? B6 pyridoxine, B6 PLP, pyridoxal phosphate. So can be connected that way. So succinyl-CoA combining with glycine, ALA synthase, vitamin B6, heme is synthesized, and it can become hemoglobin or heme proteins, any heme proteins like cytochromes and all that. Okay. The next one is malate. Malate, it will get out of TCA cycle into gluconeogenesis process, that is glucose formation. So TCA cycle and gluconeogenesis, they connected through malate. Malate is part of TCA cycle. That's how gluconeogenesis is connected. And the last one, oxaloacetate, can be made to aspartate. Aspartate is a non-essential amino acid. Aspartate. Oxaloacetate to aspartate. Okay. So these are the reactions. Means these are intermediates of TCA cycle, which can be pulled out of mitochondria to go into various other metabolic pathways. So citrate to fatty acid synthesis, alpha ketoglutarate to glutamate and GABA, succinyl-CoA to heme, malate into gluconeogenesis, oxaloacetate into aspartate.